Okay, everybody, welcome to our first Nearpod lesson. So with this lesson today, really just intended to be kind of an overview of what biopsychology is. So, biopsychology, the name of your textbook, the title of your textbook. So the focus of the textbook takes a very biopsychological approach. So we're going to just first talk about what that term means, and then we're going to look at the divisions of the nervous system. Okay, so what do we mean by biopsychology? Well, neuroscience is an important part of biopsychology, and neuroscience is the scientific study of the nervous system. And neuroscience includes under the umbrella of neuroscience, we have many different approaches to studying the nervous system. So, we have neuroanatomical approaches, neurochemistry, approaches that look at, things like neurotransmitter systems, neuroendocrinology. So that's going to look at, hormones, neuropathology. So that's going to look at, abnormal, pathologies in the brain. We've got neuropharmacology. This refers to, the effect that different drugs have on the functioning of the brain. And then we also have neurophysiology, which looks at the brain from a neurophysiological perspective, meaning that, looking at, things like electrical signaling in the brain and things like that. So biopsychology is very similar to neuroscience in the sense that it does, look at the brain from all these different approaches that neuroscience looks at it from. But biopsychology is a scientific study of biology and behavior, and the biology of behavior. So with biopsychology, this is a field that uses all of these neuroscientific approaches that I just listed in the previous slide, but it uses them to study psychological phenomena. So, with biopsychology, this is a discipline of neuroscience that integrates these various different approaches to understand how the brain produces behavior and to look at psychological phenomena, things like learning, memory, motivation, perception, things like that. So that is going to be the focus of, this class, right? So how the brain produces behavior and looking at all these different psychological phenomena by, looking at them from, these different approaches, these different neuroscientific approaches. So what are we going to be talking about? Well, we're going to be talking about the nervous system. But we're going to be talking about the central nervous system in this course. And, obviously, the brain, very important for this class. But we can divide the entire nervous system into two components. We've got the central nervous system that's very important for this course. We've also got the peripheral nervous system. So the central nervous system, this is comprised of the brain and the spinal cord. And like I said, this is what we're going to mostly care about in this class. But the brain doesn't function in isolation. So the brain depends on the peripheral nervous system in order to be able to, take in sensory stimuli and then interact with the world. So the brain doesn't function in isolation, so it's important to know a bit about these other parts of the nervous system. And for all its power, the brain really does still depend on the peripheral nervous system for, perception of the outside world and to be able to tell the body to carry out its commands. So the brain sends commands down to the body, so the brain is dependent on the body to carry out these commands. So the role of the peripheral nervous system is to carry sensory information from the body to the spinal cord and then up to the brain, and then also to bring back to the body those commands for appropriate responses. So the peripheral nervous system, kind of this one, guess if I can get my cursor to work. This other half over here, the peripheral nervous system. The peripheral nervous system, this contains all of the nerves that exit the brain and the spinal cord. And the peripheral nervous system can be divided into two other systems, those being the somatic nervous system and the autonomic nervous system. So your heart, your lungs, your digestive system, and other organs are commanded by the autonomic nervous system. And the autonomic nervous system so the autonomic nervous system is, participates in a large number of really critical regulatory functions. So things like blood circulation, secretion digestion, so secretion of digestive fluids and digestion itself. Many reflexive behaviors are carried out, with the assistance of the autonomic nervous system. So these reflexes include things like respiration, pupil dilation, sneezing, coughing, swallowing, vomiting responses. So you might think of this system as kind of the automatic or kind of cruise control nervous system because it manages many of these vital functions without conscious effort and without conscious awareness. Okay, so I should have skipped ahead here. The somatic nervous system, this is, your regulatory, those regulatory and, reflexive functions that are largely unconscious. These, things like respiration, pupil dilation, coughing, swallowing, vomiting. So these kind of automatic functions that happen, that without conscious effort and outside of our conscious awareness. So the autonomic nervous system This one is further divided. So this one here is further divided into the sympathetic nervous system and the parasympathetic nervous system. So let's look at these functions of these two subdivisions. So the sympathetic, starting with the sympathetic nervous system. So the autonomic nervous system, like I said, divided into the sympathetic and parasympathetic nervous systems. And the sympathetic and parasympathetic nervous systems, they transmit signals or commands to the glands and organs from the central nervous system, so from the brain and spinal cord. So the sympathetic nervous system activation. So this one is important for a lot of things in psychology, because the sympathetic nervous system is involved in the stress response. So this is that fight or flight system that you may have heard about in your intro class. So this is controlled by the sympathetic nervous system. So it's involved in arousal and the stress response. So when we are faced with a threat, the sympathetic nervous system is kind of like the get up and go system. It manages arousal, and it kind of helps our body cope with emergencies by preparing the body for action. So during times of sympathetic nervous system activation, our body starts to expend rather than store energy. Obviously, if we're faced with a threat, our body is going to do a number of things to prepare our body to deal with the threat, either fighting the threat or fleeing from it. So the body is expending rather than storing energy during this type of when the sympathetic nervous system is activated. Now, obviously, the sympathetic nervous system, it can't run continuously, or the body would run out of resources. So this is where the job of the parasympathetic nervous system comes into play. And the parasympathetic nervous system essentially does the opposite of the sympathetic nervous system. So the job of the sympathetic nervous system is to provide rest, repair, and energy storage. So the parasympathetic nervous system can be thought of as the rest and digest system, as opposed to the flight or fight system, which is what we sometimes refer to the sympathetic nervous system as. These two systems, usually have opposite effects on the same set of organs. So we usually view them as working like a toggle switch, so turning on one system inhibits the other. It's difficult to imagine being aroused and resting at the same time. So they're kind of like, the on-off system. Okay, so let's move on to our next part of the nervous system, the central nervous system. So we've got two parts going on here. We've got the spine and we've got the brain. So the spine, is certainly a complex organ, a complex system, contains 1 billion nerve cells. And the spinal cord is important because it delivers those signals to the body. Those commands coming down from the brain to the body, so that we can move our body. So the spinal cord is connected to the brain by a region referred to as the brainstem. So I've got that circled right here. And the brainstem is a really important region because, not only are those motor commands sent, from the brain to the brainstem and onto the spinal cord and then the body, but also those vital functions, controlling respiration, heart rate. So things like that are those signals cross through the brainstem. And, damage to these connections can have very dire consequences because we're talking about vital functions. So, your heart doesn't beat, in isolation from your central nervous system. Your respiration doesn't occur in isolation from your central nervous system. So all of these vital functions are dependent on regions in the brainstem. Okay, so let's move on to kind of the star of the show, which is of course the brain. So the brain, so people kind of, so what do we have here? We've got basically 3 pounds of squishy substance. Um, and 3 pounds of a squishy substance in humans. Um, and, looks quite different, across different species. So I've got on the slide, the brains of some different species. So the brain, some, important differences, but very many similarities between the brains of many different species. So we've got a human, we've got a chimpanzee, we've got a baboon, we've got a mandrill, we've got a macaque monkey, we've got a bear. So differences in size for sure. And differences in, the number of folds, but we do have some similarities. We've got a left and a right side. We've, all these brains have, some degree of foldiness to them. So the brain looks similar across many different species, but there are some variations in size across different species. So, and this is something that, looking at the brain, kind of people knew what the brain looked like long before they had any idea of what it did. So you think about very early in human history, hunters, probably noticed, that all animals have a brain. And they probably noticed that the brain of all these different animals, including humans, varying greatly in size, but they do look they look quite similar. So our brain contains many more neurons than the spine does. So the brain contains approximately 86 billion nerve cells. And these 86 billion nerve cells, and these nerve cells make about 15 trillion connections. So that's a remarkable kind of unimaginable number of connections. So where the spine contains, about 1 billion nerve cells, the brain contains 86 billion. So all of this, these 86 billion nerve cells, 15 trillion connections, this is all contained within this 3 pounds of jello-like tissue. So the most kind of conspicuous outer feature of the brain consists of this crinkled tissue that that we probably think of when we think of the brain. So the outer this is the outer surface, and this is called the cortex. So the word cortex is comes from the Latin word for bark. So this outer layer, the cerebral cortex, often just referred to as the cortex, named as named after the Latin word for bark. Kind of, appropriately chosen because the cortex's folded appearance resembles the bark of a tree, and also because the tissue of the cortex covers most of the rest of the brain, just like the bark covers a tree. So the brain does have many folds, um, and does have a bark-like appearance. So another thing that we can notice, just kind of upon gross inspection of the brain, is that it has two relatively symmetrical halves. So these halves are referred to as hemispheres. So we've got a right hemisphere and we've got a left hemisphere. So just as your body is, symmetrical, having two arms, two legs, and so on, so is the brain. We've got a right side, we've got a left side. Okay, so let's move on and look at some of these 86 billion nerve cells. Okay, so like all other living tissues, the nervous system is no different in the sense that it's made up of cells. So the nervous system is composed of two main classes of cells. So we've got neurons and we've got glia cells. So neurons are the basic signaling cells that transmit information throughout the nervous system. So neurons are the cells that send those electrical signals, send those action potentials, release those neurotransmitters. Glial cells, on the other hand, these are non-neural cells, but they serve a number of very important supportive functions. And many of these supportive functions are only now kind of becoming understood as researchers start to investigate glial cells further. But these cells, they provide structural support and electrical insulation to neurons, and glial cells do a number of other things, such as delivering nutrients to neurons. We'll talk a little bit more in depth about these next chapter, in chapter 3. But this is just kind of intended to give you kind of the broad strokes. So we're going to run through next the parts of neurons and in general what they do. So this should largely be a review from your intro class, but we'll just kind of run through the basic parts and what they do. So neurons are tiny, discrete information processing units. So they're processing units because they receive input from other cells, they integrate these inputs, and they distribute this processed information onto their neighbors, so onto other neurons. Neurons are arranged in networks, and these networks vary quite a lot in terms of their complexity. And, they can be simple, they can be very complex. And these networks or circuits define and control all of our behaviors and abilities, from the simplest reflex to the most complex intellectual processes. So when we're looking at neurons and the parts of neurons, we're looking at one for illustrative purposes, but keep in mind that we've got at least 86 billion of these cells. So let's start looking at the parts of neurons and what they do. So we've got first the cell body. So the cell body, it contains a number of really key structures. So it contains a nucleus, which is the spherical structure here that contains a DNA. It also contains a mitochondria. So those organelles, so the mitochondria, organelles that provide the metabolic energy for all of the neuron's cellular processes. But the key thing to know about the cell body, is, you don't have to know, what the mitochondria does, what the nucleus does. The key thing that we need to know for this class about the cell body is that it's an integration zone. So information received from other neurons, it is received through these little extensions off the cell body, which we'll get to in a minute. So incoming signals from other cells are received by these branch-like structures, and then they are integrated in the cell body. So the cell body integrates these signals that come in from its neighboring cells, and this is what determines if the neuron will send a signal of its own down its axon. So the cell body, an integration zone that combines information from its neighbors. And if this combined information, being electrical charges, so if these electrical charges reach a threshold, this will be what determines if a signal is sent down the axon. We will talk a lot more in a lot more depth about these processes in chapter 4. As far as, what do we mean by integrating signals? What are these signals? All those things, if you have those questions, answers to those questions are coming in chapter 4. Just want to cover the basics right now. Okay, next key part of neurons, the dendrites. So these are those branch-like extensions coming off the cell body that receive those signals from neighboring cells. So dendrites, so they are the input zone for a cell. So they're the input zone, and dendrites, they can take on many varied and complex forms. So this is a pretty simple kind of dendritic arrangement that we've got in this cell, but they can be really complex. So let's just skip ahead and kind of see. So this is an example of what a dendrite in the cerebellum might look like. And basically, the complexity of the dendritic arrangements for a given cell depends on, the type of cell and the location of the neuron in the brain. So some cells in the cerebellum do, in fact, look like this because of the types of functions that the cerebellum is involved in. Dendritic arrangements vary in complexity depending on the type of cell we're talking about and the type of neuron we're talking about and its location and what job it has to perform. Okay, next we have the axon. So the axon is it is referred to as a long process. So a process is just, an extension, basically. So it's a long extension uh, coming off of the cell body. And the axon is the structure that carries those action potentials that you probably learned about in your intro class, carries those action potentials from the cell body down to the axon terminals, so the end of the neuron. So this is the input zone, this is the output zone. So the axon carries those signals down to the output zone. So axons carry the electrical charge away from the cell body. And again, we'll talk more about this electrical charge, how it's initiated, how it's carried down the axon in chapter 4. Okay. One thing that's important to note is that these kind of two parts that we've just looked at, the dendrites and the axon, these features are special to neurons. So neurons, unlike other cells, have these physiological properties that allow them to transmit and process information rapidly. So dendrites and axons are features that are special to neurons and allows neurons to send signals on to other neurons. So other types of cells don't have these features. Okay, let's keep moving our way down this neuron. Next, we've got the axon collaterals. So the axon collaterals are just these little endpoints of the neuron here, these kind of side branches of the axon. And these side branches allow a neuron to send its signals to more than one neighboring neuron. Can kind of think of it, getting a bigger bang for your buck. You send one signal down the axon and it can be split up into, a few different other signaling units. Now, again, the number of axon collaterals can vary for the neuron. And it can also change depending on how many signals the neuron is sending to its neighbors. So say there's a lot of communication between this neuron and its neighbors, then perhaps this neuron will come to have more axon collaterals if it's communicating with its neighbors more often. If it's communicating with its neighbors not a lot, then there could be fewer axon collaterals. Okay, so next we have another key part of the neuron, which is the myelin sheath. So many axons are coated with myelin sheath, which is a fatty substance that acts as insulation for the axon. So this insulation improves the conduction ability of the neuron. It works much in the same way that plastic coating on electrical wires works to improve the conduction of electrical signals down a cord. So myelin sheath works in a similar type of way to improve conduction, the speed at which those signals are sent. So when we're talking about the myelin sheath, we also have to know that all along the axon, there are tiny gaps in the myelin sheath where the axon is exposed. So you can see So these are the myelin sheath segments. And then in between these segments, you can see these little gaps. These are called nodes of Ranvier. So these gaps also work to improve conduction, essentially amplifying the electrical signal and making those signals faster because the signal can jump from gap to gap to gap to gap. And this is faster because they can jump from gap to gap instead of traveling the length of the axon. So you can kind of think about it as like hopping from point to point to point, getting you there faster than walking step by step by step. So those nodes of Ranvier work in the same type of way, allowing that signal to hop from segment to segment to segment instead of having to travel down the whole thing. Okay, so now we've got the final endpoint of our neuron, the axon terminal. So the axon terminal, so this is the endpoint. This is the output zone for a neuron. Now, the axon terminal has a few different names. Sometimes they are referred to as the synaptic buttons or just simply terminals. But whatever name we're calling them, they are kind of small swellings that are found at the, absolute end point of axons. They are the sites where one neuron synapses with another neuron. And it's also where neurotransmitters are stored. So neurotransmitters are, if you remember from your intro class, they are chemicals that are released from the axon terminals, and these neurotransmitters provide the chemical signaling that allows neurons to communicate with each other through these synapses. So the axon terminal, this is the output zone. This is where the neuron's activity is transmitted across the synapse and on to other neurons. So let's look at another key feature when we're talking about neurons, because we have to know another key feature, the synapse. So synapses are the spaces between neurons where neurotransmitters are released. So we will learn a lot more about synapses in chapter 4, because it's so much more than just a space between neurons. But for now, that's kind of what we can think of it as. So we've got the space between neurons where the neurotransmitters are released. So neurons don't touch each other, right? So one electrical signal, the electrical signals that are sent down the axon, that go from the cell body down the axon and to the axon terminal, that is a signal that happens within the neuron. So that is the neuron's way of communicating with itself, basically. The synapse, this is how neurons communicate with their neighbors by, if we get enough of a strong enough signal generated, through combined inputs in the cell body, if there's enough of those combined inputs to reach a threshold, this will trigger a signal that action potential will be sent down the axon, and this will trigger the release of neurotransmitters out into the synapse. So that's what we have here. So you can think about this part right here as being the axon terminal of this neuron, and this part right here being the dendrites of the receiving neuron. So this neuron is going to send a signal down its axon. That's going to trigger neurotransmitters to be released into this synapse, those gaps between this neuron and its neighbors. And then those neurotransmitters will be absorbed by the dendrites of the receiving of the neighboring cell. And this is going to change the charge. So when the neurotransmitter molecules attach to the dendrites on the receiving cell, it's going to change the charge of that cell. And this change in charge, this is what the cell body gets. Okay, so let's just kind of look at, because this is sometimes referred to as a presynaptic and postsynaptic cell. Okay, so this would be the presynaptic cell. The presynaptic cell is the cell releasing the neurotransmitters, and the postsynaptic cell is the cell receiving the neurotransmitters. So this is kind of an important terminology to know because it's used a lot when you're discussing, when we're talking about all the different behaviors we'll be looking at. We'll often see these terms come up: presynaptic and postsynaptic. So information is transmitted from the axon of the presynaptic neuron to the receptive surface of the postsynaptic neuron through the synapse. So the synapse, the gap between neurons, and whether or not a neuron is considered to be presynaptic or postsynaptic simply depends on where it is located in the chain of events. So if we have our neuron that we've been looking at all along, we can call this one the presynaptic cell because it's the one sending the signal down its axon and releasing the neurotransmitters from its axon terminal. And then these neurotransmitters are going to attach onto the dendrites of this postsynaptic cell. So it's only called the postsynaptic cell because it's just second in the chain of events. So if we were to add on another cell into our chain, we would now be referring to this one as the presynaptic cell and this one as the postsynaptic cell. So as you can see, it just depends on where the cell is at in the chain of events, whether it is sending the action potential or whether it is absorbing the neurotransmitters. So back to our synapses. So again, this is the spaces between cells or between neurons where the neurotransmitters are released into. And here we have what is called the presynaptic membrane. Again, so this is just, because it's the cell that's sending the signal, it's presynaptic. And so this is the very edge, the very end of the terminal, called the presynaptic membrane. And this is called the cleft. So that space right here where the neurotransmitters are floating around, this is called the cleft. And this part right here, this is referred to as the postsynaptic membrane because it is receiving those neurotransmitter molecules. Okay, so now that we know a little bit more, or we've at least refreshed ourselves from our intro class, let's look at another important topic that comes up in this course. So we've got the use of human and non-human research subjects. So a lot of the research that we talk about in this course will be based on research that has used human and non-human subjects. So let's look at some of the advantages of So let's look at the advantages of both types of research subjects. So humans are great research subjects, for a number of reasons. Probably the most important being at the end of the day, we are interested in, uh, knowing what the human brain does. Um, and also, humans can follow directions, right? So they can report a subjective experience. Um, they are less expensive in the sense that, you don't have to house them, you don't have to care for them. They can come in, do their research study, and then go home and take care of themselves. So that's certainly, a couple of the features, following directions and things like that. You don't have to spend extensive amounts of time training a human to do, a simple task, a simple research task. And also, of course, human subjects have a human brain, which is what we are ultimately interested in. So, and those are all definitely good features. But there's a couple of downfalls with using humans as research subjects. So one, and this leads us to our advantages of using non-human subjects. So one key advantage is that they have there are many non-human subjects that have much simpler nervous systems. So we'll look at an example of this. I've got a video to show you looking at habituation in the sea slug. Kind of a nice way to illustrate how a simpler nervous system, being able to look at a simpler nervous system, just makes things a little bit more manageable, because when very little is known about something, it's best to start small and simple, right? So many non-human subjects have much simpler nervous systems than a human. And also, we can also look at comparative approaches. Allowing for, some comparisons between species, and also, and this is kind of a key one as well, with non-human subjects, there are fewer ethical constraints. There are just, things that you can do with non-human subjects that you just can't do with humans because it's not ethical. Of course, there are ethical guidelines that have to be followed for non-human research subjects, but there are fewer ethical constraints for non-human research subjects. And also allowing, for more experimental control over the animals' life experiences. Eliminates confounding variables, a term that you should remember from your intro. Okay, so how do researchers study the brain? So let's just do kind of a quick overview of some of the methods that are used to study the brain. So these are some of the methodologies that will come up, and certainly we'll get more in depth into methodology when we cover chapter 5. But let's look first at experiments. Of course, experiments are the gold standard of research because experiments allow researchers to determine cause and effect relationships. We can have different types of experimental designs. So we can have something called a between subjects design. So this is where, you have two research groups. You've got two groups of subjects. They are tested under different treatment conditions. And then you can look and see what your experimental manipulation, so one group will have received the experimental manipulation, and then you can look and see what the outcome is. So for example, say you're testing a new drug, so you've got, group A and group B. Group A is getting the drug, group B is getting a sugar pill, a placebo pill. So that's an example of a between subjects design. And you can establish, so if you're testing your drug, you want to see if it's having an effect. You can do this with a between subjects design where you've got, group A receiving the drug, group B is not. So you can give group A the drug, see, whatever you're measuring, what the outcome is, and compare that with the group that didn't get the drug. So that allows you to determine a cause and effect relationship. You can say, after running an experiment like that, if your drug is causing that effect that you're measuring. So we can also have within subjects design, and this is where the same group of subjects are tested under different treatment conditions. Whenever we're having you doing an experiment, we're going to have independent variables as well. So the independent variable, these are the things that you are manipulating in the experiment. So if we come back to our drug example, the drug would be the independent variable. So that's the thing that you're manipulating and saying group A is getting it, group B is not. And then you're going to measure outcome. So say we're talking about depression. So group A is getting the antidepressant, group B is not. And this brings us to our dependent variable. This is the thing we're going to be measuring. So the dependent variable would be, say you're talking about depression, you give your subjects a depression questionnaire. So they get their antidepressant or they don't, and both groups fill out that depression inventory, and you are the dependent variable would be their score on that depression questionnaire. So that sounds all well and good, but experiments do have these unfortunate little things called confounding variables. And these are, these are unintended differences between conditions or subjects that can influence that dependent variable, that thing you are measuring. So usually with experiments, researchers try their best to minimize confounding variables, but nothing's perfect. It's very difficult in practice to make sure that there is only one difference between the conditions, right? So again, if we come back to our drug example, you get group A getting the drug, you got group B not getting the drug. So that's one difference, right? Whether or not the groups are getting the drug. And that's the difference that you're interested in. But as you can imagine, if you've got a group of 20 people in group A and a group of 20 people in group B, there's going to be a lot of other differences other than just that whether or not they got the drug, right? So these confounds are differences that the subjects are bringing into the experiment. It's not possible for the researcher to know what effects the confounding variables are having. Difficult to measure confounding variables. Try to minimize them, but it's very difficult to completely minimize them. So full-on experiments are great. Like I said, the gold standard, but sometimes depending on what you are interested in researching, it's not possible to conduct a controlled experiment. So then we have these quasi experimental designs. And in these types of designs, these are often used, for kind of when researchers are looking at, kind of real world situations. Um, uh, you know, so um, so let's take an example of this. If you're looking at, say, for example, excessive alcohol drinking and memory impairments. So here, we can certainly look at this, but in this type of real world situation, what we have is research subjects who have self-selected themselves into a specific condition, right? So excessive alcohol intake or not. So they have essentially self-selected themselves into these conditions. These subjects in the excessive alcohol intake group have, assigned themselves to the treatment condition. Because of course you can't, make people drink excessive amounts of alcohol and see what it does to their memory, because that's not ethical. But we do have people who decide to drink excessive amounts of alcohol on their own. And you can, look at memory impairments in that group. But the problem is that they are self-selecting. And this means that the researcher can't control for potential confounding variables. So because of this, it doesn't allow for direct establishment of cause and effect relationships. And also many, many more confounding variables. Say the excessive alcohol intake group, have some genetic differences, perhaps some lifestyle differences, and all of these things are going to contribute potentially to memory impairments above and beyond the excessive alcohol intake. So quasi-experimental studies certainly necessary, when controlled experiments are not possible. Okay, we also have case studies. So case studies are used when we have rare types of conditions that maybe you only come across a few people that have it. So case studies are studies that focus on a single subject. So we will learn later in the course, we'll talk about case studies throughout this course. So HM, right? So HM, the famous amnesiac subject who lost his long-term memory. This would be an example of a case study. And it's an in-depth study of one single person or a handful of people. And certainly, issue of generalizability, right? Because you're talking about one person with a rare condition. Certainly, when you think about HM definitely contributed a great deal to our understanding of the neural basis of memory. But it's still difficult to generalize from people with rare conditions to the general population. Okay, we also have physiological methods. So these are methods that where the researcher kind of directly manipulates the nervous system. In some way, in a controlled laboratory setting. So for example, lesioning studies where the researcher intentionally damages a part of an animal's brain. Or when we are directly recording from individual neurons in an animal. These would be physiological methods. For these types of physiological methods, we're talking mostly about animal research subjects in these methods, not humans. Okay, pharmacological methods. So these ones involve, it's similar to physiological methods, except that instead of, manipulating the nervous system through something like lesions, instead we would be manipulating the nervous system pharmacologically, so with drugs. So, these types of methods looking at the effect that different drugs have on various aspects of behavior. Neuropsychological methods. So, these methods focus on the effects of particular brain dysfunctions. So, whether you're talking about, you know, uh, accidental, brain damage, um, you know, uh, uh, disease, things like that. So, with these types of neuropsychological methods, usually we're talking about case studies, um, and this type of method would be involving more applied, applied research, to facilitate diagnoses, treatment, things like that. Okay, psychophysiological methods. So these methods look at the relationship between physiological and psychological processes um, in humans. Um, so psychophysiological methods, it could be, a number of different things. Um, Looking at EEG, so the electroencephalogram, where we place electrodes in the scalp and measure the electrical potential of the entire brain. So this is a psychophysical method, because you're looking at physiological activity in the form of the electrical potential of the brain and its relationship to some psychological process. So, for example, looking at pictures of inanimate objects versus pictures of human faces. So these methods are non-invasive and can be done on humans. And there can be a number of other things as well. So muscle tension, so measuring muscle tension of muscle responses in your muscles, measuring pupil dilation, electrical conductance of the skin, right? So this is a good one. And this is also electrical conductance of the skin. So this can be used to measure the stress response because basically the more sweating you do, the more stressed you are. So that can be measured just simply by, using the galvanic skin response, just measuring the amount of sweat on your skin. So again, a very non-invasive, easy type of method that can be used on humans, most importantly. So other types of psychophysiological methods, eye tracking. So this one, I've got a video to watch on this one. But first, what is it? So eye tracking just simply involves tracking the movements of the eye. So for example, person can be set up with this camera that tracks the movement of their eyes. So, and you can sit them in front of a screen and get them to like read some text, right? And what this device will do, it will measure the movement of their eyes as they read the text. So I've got a good video on this, looking at eye tracking, shows you how the technique works and what it can tell us. So we'll watch that video. Okay, another way that we can study the brain using imaging. So MRI being one of the most popular ones. So um, this was a big step too. Different types of neuroimaging because you know up until kind of the late 1970s, most of our understanding of the structure and function of the brain came from post-mortem human specimens, or was inferred from animal studies. So these would, so post-mortem being, autopsies on deceased people. So with something like MRI, this, allowed researchers to measure the functioning, structure and functioning of the living human brain. And it's based on the fact that tissues behave differently when they're exposed to a magnetic field, depending on what they're made of. So tissues with greater fat content will appear white, and tissues with less fat will show up as dark. This and these differences in how tissues how they behave when they're exposed to a magnetic field. This allows us to get a really clear picture of all these structures in the brain, which we will learn about in chapter 3. And we also have kind of another advancement in the MRI technique being fMRI, functional MRI. And this allowed researchers to measure blood flow in the living, functioning, behaving human brain. And it's based on the concept that brain regions that are more active, brain regions that are working hard, require more blood. So we'll learn a lot more about these techniques in chapter 5. So we've got all these different techniques, and certainly this isn't an exhaustive list, but, we've got a lot of different techniques. And it's a good thing we do because each technique has its own weakness. Each area has its own weakness. And whether you're using, whether you're working in a research area that is using, physiological methods or, psychopharmacological methods, imaging methods. Each area has a weakness, right? So this means that, in order to develop good questions and get good answers, there has to be collaboration and, different approaches used, so that each approach can compensate for the shortcomings of the other. Okay, so one more very quick but very important topic: research ethics and biopsychology. So we do have, all universities and research institutions have mechanisms in place to protect human participants and animal research subjects. So, we have things like university review boards, institutional review boards that are, armed with sets of guidelines and rules and regulations to protect research subjects, both human and animal. And, this is particularly important because, especially in a course like this, we will be covering a lot of research that uses animal research subjects. And this can be uncomfortable, for a lot of people. If you're an animal lover, some of it is, some of it can be disturbing for some people. So it's important to remember that animal research has contributed so significantly to our current understanding of the brain and of behavior, neurological disorders, among other things. So certainly animal research has made so many important contributions, but it does come at a great cost to those animal subjects. You know, many of them having to be sacrificed in order to complete the research. So, so as we go through this class, this course, and we look at all the different research, that is out there, a lot of it does involve animal subjects, and a lot of it does involve harm coming to them in the interest of the research. But this is very regulated by university review boards and institutional review boards, and there are very strict Canada-wide guidelines for, ethical considerations when using animals as research subjects. And certainly if there is harm coming to the animal, there has to be justification, extensive justification for that. So keep that in mind as we go through the course, that when animals are used as research subjects, it is not just people doing whatever they want. It is tightly controlled with guidelines in place. And so yeah, keep that in mind, contributing significantly, but it does come at a great cost to animal subjects. Okay, so that's the end of the lesson, and we will pick up for a live class on Tuesday.